Hi everyone, and thank you for inviting me to talk today. My name is Ursula Delaney, Committee Member of SCN2A Australia and mother to Eva with this rare genetic disease. Today I'm going to talk about the challenges, the heartache, the roadblocks and the hope. SCN2A is a debilitating genetic condition. A quick parent synopsis of the disease would be there are various presentations in SCN2A. This condition can be benign, mild, moderate, and severe. It comes with many comorbidities: seizures, no seizures, movement disorders, autism, and possible death. My daughter Eva was born in December 2015. From her birth, I very quickly learnt our hospital system was flawed. For the first couple of days, Eva would not feed. She would sleep constantly, and when awake, she'd make these strange movements, go red in the face, and scream. I constantly raised this with doctors and nurses to be very quickly dismissed about being a first-time mum. Finally, one of these events actually occurred in front of one of the nurses. The nurse believed she was having seizures, however, the paediatrician actually disagreed. On Christmas Day, Eva was transferred to our local children's hospital. We underwent an EEG and some other testing. We went home with the diagnosis of Sandifer syndrome, which was an extreme reflux condition that looked like seizures. After a week of being at home, we actually ended up back in hospital with Eva in status, so one seizure after another. This is when our lives truly came crashing down. Medication after medication, and nothing was stopping them. We felt like we were actually about to lose our child. She was laying there basically lifeless. After a very long admission, we were sent home with a child still having uncontrolled seizures. We were sent home with zero support and no idea what to do or what to expect. It was absolutely devastating as a first-time mother to be woken by a child every 20 minutes having seizures. We went in and out of hospital with no answers as to why Eva was so unwell. At 7 weeks old, Eva was overdosed, receiving 200 milligrams of a drug instead of 20. As a parent, you go through so many emotions with a child who is unwell. You have to grieve not having a normal child. You have to adjust your perspective of what makes you happy in life. It changes and shapes every part of you as a person. At 5 months old, Eva's brain shrank and she suffered a brain bleed. In Australia, if your child has a bleed on the brain, you're instantly investigated for child abuse. I was subjected to days of questioning, and Eva had to have x-rays of her entire sick body to make sure we didn't harm her. As a parent, I was absolutely furious by this. There was quite clearly something wrong with Eva from birth, so why were they subjecting us to this? The hospital decided I should go and discuss how I was feeling with a clinical psychiatrist. I'll never forget this moment. She started talking, and part of her opening conversation was, as a parent in the system, there are two types of mums, a good mum and a bad mum. She then went on to say that a good mum listens and does what the doctors tell them to do, and a bad mum doesn't. After this conversation, I realized I am the bad mum, and so I am questioning why things are being done the way they are, and I'm calling them out on their mistakes. It was that moment in time that I promised myself I never want another parent to be made to feel as low as I did, and I have to fight. And if I'm going to be a bad mum to do that, to get the treatment that Eva deserves, that's exactly what I'm going to do. My first step was sitting on a board to bridge the gap in genetic epilepsy. I've spent the last couple of years helping to rewrite policy on the treatment of these kids. I realize that we can write as many policies and procedures as you like, but when it comes down to the mentality of these doctors, nurses, and the people who work in our system, it takes them to want to change. And I'm hoping that by speaking about this, we can create change. At 6 months old, Eva received the diagnosis of SCN2A, and that's when I was off, reading hours a day about the disease, setting 2 laptops up side by side, reading medical journals, publications, Googling words so I understood what they meant. I realised the drug they'd overdosed Eva with at 7 weeks old was likely going to help control her seizures more effectively. They removed the drug on the first day of trying it and overdosing her out of bad luck. I took Eva to see a neurosurgeon to discuss her MRI and the bleed. This neurosurgeon was the head of the Australian Medical Association at that time. We spoke to him about the issues that we'd had to date with the medical system. He then connected me to a neurologist he thought may be able to help. This neurologist knew the disease, and he was a man that was so humble and so kind. I knew I wanted to work with him, and we worked together towards better seizure control. Once we achieved this, however, we didn't realise that by doing this, we were awakening a child who was in serious neuropathic pain and had an underlying movement disorder. As a mother, I went from being woken up every 20 minutes from seizures to a child who wouldn't sleep any more than 3 hours a day. I was literally exhausted. On top of this, I was also running a property management company. I would sleep on the floor in Eva's room every single night next to her, constantly arching and twisting her little body in pain. I found myself laying there at night thinking in my mind, do I want a child to have seizures and sleep, or a child awake and in pain? I actually found myself wishing seizures. I had to learn not to focus on the past and to focus on the future, not to dwell in hospital admissions that went wrong or doctors that won't listen to you, to stand up, be strong, advocate for your child, and remember to still love them for them. Be happy they are with you. I've watched Eva fight for her life so many times. She's had so many surgeries on her little body. She fights to be here, and I know I need to do the same. Sometimes I feel like it's her teaching me to be the advocate. I feel like if she could talk, she'd say, "Mum, get your shit together. We've got this." At 2 years old, Eva's father left and she became extremely unwell again. This time going to sleep and not waking up for weeks. An EEG revealed the hips arrhythmia had returned and she was in an extremely bad way. During this hospital admission, my mum actually called me. She said she'd been Googling SCN2A and she found a company that was looking at treatments for kids like Eva. She sent me the link to register for more information, which I did. Kelly from RogCon called me. She explained that it was in early stages in learning about the gene and potentially creating treatments for our children. She spoke to me about the scientific works that were being done out of the Florey Institute in Melbourne. I couldn't believe what she was talking about. Kelly and RogCon were based in the States, and it was actually happening here. Kelly arranged for a skin biopsy of Eva to be sent to the lab to be studied. RogCon was a biotech company that was set up by two incredible parents, Alex and Kelly, who had children with SCN2A and were trying to create novel therapies for our kids. I was so incredibly excited by finding this. I decided to share this information within an SCN2A Facebook group. My post was removed by them. I was so shocked. I naively thought that they just didn't know about this information. I was at this point a little bit confused about their actions, so I decided to fly myself to Melbourne with Eva, meet the team involved. I met with Dr. Catherine Howe, who was conducting the natural history study for our gene, and then went to the Florey Institute. I was looking at rats with SCN2A receiving treatments, brains in a dish, and EEGs on live cells. I have zero science background and I didn't want to leave. It was so exciting seeing everything that was being done. It was this point in time that I realised an SCN2A group based out of Australia was not going to represent our children here, and trying to fly sick children like Eva to another country for treatment would be almost impossible. This is when I joined SCN2A Australia, which is led by Chris Pearce, a panellist here today. We both had the same mission to obtain better healthcare for our children and working towards better treatments. We both felt it was so important to share this information back to our community. SCN2A Australia has done this through some incredible podcasts and webinar conferences. We forged forward to make some amazing relationships with some critical professionals and industry bodies in the SCN2A world. I was so thankful to be under the wing of Chris, who not only ran SCN2A Australia, but she was also one of the leaders of GETA, Genetic Epilepsy Australia. I was excited to hit the road with Chris. Chris's husband actually called us Bonnie and Clyde. I feel so incredibly honoured to work with Chris and learn from her. We have attended genetic epilepsy roundtables, Parliament House, rallying for better treatments for rare disease. We've been lobbying all levels of government to highlight the needs for our families. The road getting to clinical trial is long and it's hard. It requires researchers to be interested in your gene, industry to want to start looking at precision medicine for your gene, and strong advocates. ROGCON was formed in 2015 and was led by two SCN2A parents and an Australian neuroscientist, Dr. Stephen Petro. In 2017, ROGCON engaged Ionis Pharmaceuticals, the world leader in discovering RNA-targeted therapeutics, in collaboration to seek an ASO treatment that will target SCN2A mRNA treatments. From there, in 2019, ROGCON, Ionis, and Praxis Precision Medicine entered into a three-way collaboration around RC222, the SCN2A ASO program. From 2019 to today, Prax 222, the SCN2A ASO program, originally called RC222, is currently undergoing evaluation in late-stage IND-enabling toxicology studies, with the intent to file a request with regulatory agencies to dose SCN2A patients in the first half of 2022. At the moment, treatments for our children is medication that only helps the seizures or the movement disorder. These medications bring with them so many side effects like liver failure, vision loss, electrolyte dysfunction. They don't help the dysfunction of the gene itself. And this is currently what is being developed. As we lead closer to clinical trial, we realised as SCN2A Australia, we needed to start pushing for clinical trial sites in our region. This cannot be done with a group outside of our region. It had to be done by us. All the fundraising from these groups will not help us in Australia. Due to the research being founded in Australia and the natural history study also being done here, we hope we are in a position to fight for our families for access to clinical trials and potential clinical trial sites. Chris and I have been working with the clinical trial coordinators in Melbourne and Sydney to put forward these hospitals as sites for clinical trials and advocating for further awareness in our gene. We want to ensure everyone knows who we are and we want to work with them. We are currently sitting and anxiously waiting to find out when the ASO trial will commence and if Australia will have a clinical trial site. So wish us luck.